Okay, so we're going to introduce prokaryotes, which simply means the following. If it's not a virus, this is as far as we know, okay? Who knows? Maybe there's some other life form that we don't know about on this planet. If it's not a virus, it's not a eukaryote, it's a prokaryote. So if it's a cell, remember a virus is not a cell, right? If it's a cell and it has no nucleus, it's a prokaryote, okay? So this is basically bacteria. This is like your average bacteria. But there's no such thing as an average bacteria because it would be like putting up a picture of Erica and say, this is your average human. Now, that means that Erica, has obviously features in common with all of the humans, but it doesn't mean every human looks like Erica, right? So this is what an average bacteria would look like. It's it's got an outer membrane that's in front of a, what's another word for plasma membrane? Cell membrane, okay? Remember the parts of the cell? Grade 9, 8, 11, 10, whatever, 10. Okay? So a prokaryote is basically a cell that has an outer wall, a cell membrane. It's got no nucleus, so you can see that here. Okay? And we'll talk a little bit about bacteria today. I'm not going to go into too much detail because I want to focus on characteristics, okay? So here's a nice little picture. This is a picture of bacteria. This is lactobacillus. I'll teach you how we actually can name bacteria. It's a cool little picture. So you'll notice that they have like a very specific shape. Unlike viruses that have very weird shapes, they're pretty simple in terms of their shape. This is a picture of what's called a biofilm. So everybody notice after a while in your teeth you start feeling a little bit like slimy? Just before you brush them, right? You notice that they start to feel a bit of a film buildup. That's actually bacteria secreting what's called a biofilm. This is actually a picture of the bacteria in that film. Okay? That's kind of a cool picture. What do you think this is? The volcano. People walking up the volcano to their doom. What is it? A carpet. Yeah, it is an object. Yes, it is. It's magnified. Quite a bit, actually. I can't remember the exact number, but it is magnified. This is a picture of... Oh, yeah. This is why my presentations last forever. Yes.
My pillow.
Think, okay, I'll give you a hint. Think, Remembrance Day.
Poppy flowers.
Okay, but what's the thing that's attached to the poppy?
Needle.
This is the head of a pin. Okay. Now, it's color enhanced, so you can see it better. The yellow stuff is, guess what? Bacteria. Okay, so we know bacteria are extremely small. We know they're widespread. You can pretty much find them anywhere. Okay, they can survive some pretty crazy environments. You can find them in nuclear reactors. Okay, you can find them in frozen lakes. You can find them in really hot environments, really cold environments. They're everywhere. They're also the first living things on this planet. Okay, so if you go back about 3.6 billion years ago, you start to see signs of life, and the first living things would have been bacteria. Eukaryotes would have evolved later on because it's just a simple rule. Things that are simpler should evolve first. Right? And bacteria are simpler than eukaryotes. So we're going to be talking about bacteria. I'm not going to go into too much detail, so I'm going to go over like a few little things about bacteria. But we mentioned that there's actually two groups of them. There's Archaea and eubacteria, and you have a whole lab that revolves around this. Right? Oh, sorry. Yeah, you're right. Part of a lab. Okay. So here are some characteristics of prokaryotes. They don't have a nuclei. They don't have other membrane-based organelles. Like, for example, you know how the mitochondria has its own membrane? Remember some of the organelles from cell biology in grade 9 or 10? Like lysosomes, chloroplasts. They do have ribosomes. Actually, all cells have ribosomes. But they don't have a lot of the organelles that you would find in a eukaryotic cell. Okay? Yeah, they don't have the mitochondria. Very good. And that makes sense because remember the mitochondria used to be a bacteria. Right? Used to be bacteria. Okay. So, here's some other characteristics. They typically have one large chromosome. Whereas if you think of your cells, how many do you have? Yeah, 46. 23 pairs, right? Their chromosome is looped. So in other words, if it's looped, there's no end. Our chromosomes are basically like a ball of yarn, but we know a ball of yarn has a beginning and an end if you stretch it out, right? In other words, there's our loops. They also have a little piece of DNA called a plasmid, and that plasmid carries some extra genes, and I'll talk about plasmids a little bit later on. So what you might see with the bacteria is one looped chromosome and maybe several of these plasmids. Eukaryotes are typically larger, typically have many chromosomes. The chromosomes are typically, they exist in two sets. They're diploid. Remember, one set from mom, one set from dad. Okay. Bacteria also have the ability to produce what are called an endospore. And that's basically like a protective coat around the genetic material. And you can see a nice little picture here of that. So here's the bacteria. You see this section here, this zone, that's an endospore. Now, what an endospore does is, just if you put bacteria in a really harsh environment and you kill them, there is a chance that the endospore can actually protect the valuable genetic material and allow the bacteria to reform. So, you guys know about like food preservation techniques? Like stuff that gets canned, that has to go through this whole process. So you gotta make sure that you really do a good job of killing the bacteria, because if the endospores survive, you might have destroyed the cell, but they can actually come back. So it's a neat little trick that they have. A lot of bacteria have flagella, okay? But the flagella that they have is different than the one we see in eukaryotes. The one in eukaryotes actually swings back and forth like this, okay? Like a fish's tail. The bacterial flagella actually spins like a motor, okay? So it's not the same thing. So what do you call something that has evolved that looks similar but is not the same? Yeah. So the flagella we see in eukaryotes is an example of convergent evolution. This is not the same type of flagella. Okay. Here is Salmonella. Is that a famous one? Where do you get that from? Chicken, pork. Okay. Even apparently turtle eggs. Apparently turtles have they're not really the most hygienic of pets to keep. I don't know. I don't have a turtle. They do, right? Yeah. So here's a neat little picture of Salmonella. Now, what may look like flagella are these other things called pili. So pili are these things that come out of the cell, but the flagella is used for motion, right? It's used to move the bacteria. Pili are used to anchor. So they're used here to actually stick to the surface of other things, but they're also used in conjugation. Now, do you know what conjugation means? Conjugation. That's cute, guys. Thank God I teach at Moral Park. Some other schools, they may have said the word sex. Okay? So, conjugation in this case refers to the exchange of material, which would be a primitive form of sex. And we'll talk about that in a little bit. Okay? Here, so, what if you pick up a biology textbook from maybe 20, 30 years ago? I could be wrong with the dates. Well, when I was going to school, we were not taught that there were two types of bacteria. We were taught there was only one type. Okay? And it was grouped, the kingdom was called Monera, and that referred to the bacterial kingdom. Here's the thing. Since then, it's been reclassified. There's actually two groups of bacteria. Okay? Don't worry about all of this. I just want you to understand that there's two groups, that they're different enough from each other that they're considered to be their own thing, okay? So there's the regular bacteria, and then there's another group called Archaea, okay? Now, the Archaea ones, they refer to things that live in, like very harsh environments, okay? Such as, this is a picture of a very hot spring in Yellowstone, sorry. Okay? So they can live in really hot environments, or these are salt pools. They can live in very salty environments. Okay? Here is an example of one. This is Natronococcus. I'll explain what how to name bacteria in a second, okay? And it can grow in a very, very salty environment. Now, you know that salt can kill things, right? Okay, because it draws water out of organisms. But this thing can live in a very salty environment that most other things can't. Here are some examples of Archaea. Okay, halophiles, thermophiles, anaerobes. Again, I don't really care if you know the little details in this unit. The biggest thing in this unit is understanding the groups. What makes them a group? Like, what are the characteristics of the group? What makes this group different than that group? So don't worry about, oh, I gotta memorize what a halophile is. I gotta memorize I don't care about that stuff, okay? I want you to understand the big picture. Like, what are the characteristics that make this group different than that group, okay? Okay, here's just some pictures of Archaea. Now, because the main topic of this unit is classification, one of the things we'll do next week is classify bacteria, okay? And we'll be doing a neat little experiment where we're actually going to classify the bacteria based on their cell wall. So if you go back to the picture of bacteria, they have a membrane, but around the membrane is a cell wall. What do you think the purpose of the cell wall is? Yeah, very good. It's for protection, okay? And you could use the cell wall to actually classify bacteria, and I'll explain that in a minute. You can classify bacteria based on what they eat or how they get their energy, okay? You can classify them based on their shape. So, what shape is this? Yeah, circle. See, you can see that these little circles. When we do our experiment and we stain the bacteria, you should be able to see their shape. Okay? It's either going to be circular or rod shape. So remember the example I showed you first was a rod-shaped bacteria. There is another type, which is here, spiral shape. Okay, so here is circular. You can see that circular. This is rod shape, and this is a spiral. There's a way that they're named. So, like, you notice the circular ones have this name, Koki, and the rod ones are bacilli, and then the spiral one would be spirulli. And now, if you think about the name of this bacteria, it's called lactobacillus. Okay? So, if you go back, bacillus is what shape? Yeah, it's rod shape. So when you have the name of the bacteria, often, like, the name of the shape is in there. So yesterday I showed you an example of bacteria. It was called Thermococcus, right? So what shape should it be then? Should be spherical, right? Staphylococcus should be sphere. Streptococcus would be so that would be circular. Okay? So that's one way to classify bacteria, is by their shape. What we'll be doing is a neat little experiment that we can actually visualize them, and this is called a gram stain test. I'm not going to go into too much detail today, but that's what we're going to do we'll start it on Monday, but we'll finish it on Tuesday. But the gram stain test allows you to see whether or not bacteria are either going to be purple or pink when you put them through this procedure. The idea is pretty simple, actually. So this is like the membrane of what's called a gram-positive bacteria. You'll notice here's the membrane, and it has this very, very thick layer. This is a gram-negative bacteria. You'll notice that that layer, it looks different, right? So here's the membrane, and that layer is thinner. Here's the thing. The test is pretty simple. What you do is you add a chemical called crystal violet, which is purple. Okay? And if the bacteria is gram-positive, what happens is the crystal violet gets in here and gets stuck there. And you're going to be adding alcohol. Now, you guys know that alcohol can remove things, right? You can use it to clean. The alcohol cannot get rid of the crystal violet because this wall is so thick that it protects the dye. So your bacteria ends up staying purple. But if you have a gram-negative bacteria, you add the crystal violet, then you add the alcohol. But the problem is because the membrane, because this is so thin, it doesn't protect the crystal violet, so the alcohol washes it away. And then what you do is you add a chemical called safranol, which is pink. So what happens when you wash away the purple and you add the pink, the bacteria ends up becoming pink. So when you do the experiment, if the bacteria ends up being purple, you know it has a very thick wall. And if it ends up becoming pink, it has a very thin wall. Now this is important because in medicine, you want to know what bacteria you're dealing with. Okay? You want to know, because their medications will work differently on different types of bacteria. Okay? But for us, it's a neat little way to do an experiment where you can actually see them. And you can not only see their color, but you'll be able to see their shape. Right, yeah. Pink means it has a thin wall, which is gram-negative, and then purple means it ends up being thick wall, gram-positive, okay? So we've been looking at classification, and one of the things you were looking at in your lab, and this is a huge hint, but I think you already know the answer, is what are we more like? So because there's two groups of our bacteria, Archaea and bacteria, are we more like one or the other? Yeah, I think the best answer is that we're a hybrid. But here's the thing. If you were to look at most of the trees that you'll see in your books, they mostly show this kind of a relationship where we are actually more closely related to Archaea than Archaea is to bacteria. Okay? But that doesn't mean that if you looked at every gene, that it would match up with Archaea. Because you did an experiment where you took proteins, and a lot of them matched up with bacteria. Because the answer is we are a hybrid. We may be more like Archaea, as this tree suggests, but that doesn't mean we're not part bacteria too. We're a little bit of both. Okay? So, okay, this talks about the difference between bacteria, Archaea versus eukaryotes. It's summarized here. Again, you don't need to focus on that. Okay? Don't need to focus on. All this says is that when you look at it, there are. Now, I didn't mention this. You know how we talked about the groups? What's the biggest group we looked at yesterday? Kingdom, right? Well, and what kingdom did we look at? It was the animal kingdom. There is a level above kingdom called a domain. This says there's 3 domains. And there's enough difference that there's separate domains. Remember, these two used to be one domain or one group. But you can see this says there's enough differences between them that we can classify them as 3 big groups. So we have this domain, and this is the one that we'll focus on because this is the domain that includes animals, fungi, what else? Well, people are animals. Animals, fungi, thin green, plants, and there's another group too called the protists, which we'll talk about later on. Okay? Okay. How do bacteria reproduce? Asexually. So how do they evolve then if they reproduce asexually? Because in order to evolve, to change, you need to get variety. So how do we get variety in bacteria? Well, one way is through mutation, but another way is actually through some primitive form of sex or the ability to pick up genetic material. So bacteria can get variety because when they reproduce, most likely they'll make exact copies of each other, right? But sometimes we get mutations, and we know that can get, variety, that will generate variety. But they also have a few other tricks up their sleeve. They could pick up bacteria from other bacteria. Now, we can't do that. I cannot get a gene from Vanessa and put it into my genome. I can't. But bacteria can do that. It's called transformation. They have the ability to pick up DNA from other bacteria and basically change them. That's kind of cool, right? Imagine you could pick up DNA from someone else. They have the ability to pick up DNA through viruses. So when viruses, remember what viruses do, right? They attach to a cell and they inject their genome. Sometimes the genome that gets inserted brings in new genes, and the bacteria actually acquires those genes. We have a lot of genes in our genome that are inserted through viral infections, and they just basically ride along with our genome. Another way they can get new DNA or variety is through conjugation. That means that two bacteria can hook up. Oh, and you might think, well, doesn't that sound the same as transformation? Transformation could be through a dead bacteria. A bacteria can come across a dead bacteria and pick up some DNA from it. Conjugation is through two compatible bacteria. Now, there's no such thing as boy and girl in bacteria, but they would have to be compatible in some way, okay? And you see that right here. These pili can be used to actually transfer DNA. Here's a better picture, okay? So here is one bacteria. You can clearly see it's not the same as this one. It's different, but they can make a connection and they can transfer DNA to each other. No, they change. That's a good question. They can change. So, for example, if this bacteria has, let's say it has a gene that gives it antibiotic resistance, right? It could, in theory, transfer that gene to this bacteria, and then that bacteria now will become antibiotic resistant. Isn't that kind of cool? The ability to transfer information to each other. Well, they wouldn't. We got to remember there's going to be hundreds or thousands of genes. You're only transferring one or two of them. So you would be a little bit different, but you wouldn't be dramatically different. Yes? Yes, it makes a copy of itself. Yeah. Otherwise, it would be pretty. Can you imagine that? That would be pretty dumb. Just give away all its wealth, right? They would have probably many copies of that gene and send it over. Now that's important. I'll tell you why that's important. So we've been talking about the three domains, right? The eubacteria, archaea, and eukaryotes. And again, this picture shows you that archaea are more like eukaryotes than they are like eubacteria. Because remember how you read these trees, right? See the branch point? It's here. Whereas the branch point for archaea and bacteria is further back. Now, the reason why I brought this up, this slide, and the ability to exchange information is we have a clear understanding of who your ancestors are. If I said to you, who are your ancestors? You would say, well, it's my parents. It's my parents' parents. It's my parents' parents' parents. So basically your parents, your grandparents, your great-grandparents, and so on, right? And you could, they have all those websites you can trace your ancestry, right? Bacteria, it's very hard to do that because of one big, huge difference. We can't do this. You get your genes from your parents, who got their genes from their parents, who got their genes from their parents' parents. Bacteria can get genes from people that aren't their parents. So here's the thing. If you want to know who, the common ancestor is of you and your brother, that's easy. It's your parents. But when you do that for things like bacteria, you can't. Because they can mix up their genes. They can swap. And you don't know where that particular gene came from. So imagine we were sequencing the genome of this bacteria, and we came across that antibiotic-resistant gene. Now here's the thing. You would have assumed that it would have come from his parent, right? But guess what? Maybe it came from this cell. So when we're trying to, and that's actually one of the ideas in your lab, right? Trying to figure out what is the common ancestor of the eukaryotic cell. The answer is we can't know. Okay? The reason why we can't know is because of this. Now what do you think this means? Lateral gene exchange or lateral gene transfer. What does lateral mean? Side to side. Side to side. We get vertical transmission. It's, us, we got it from our parents, further back in time. We got it from their parents, further back in time, right? Lateral transmission is something like this, where bacteria can get together and exchange DNA. And if that's the case, if they're doing this, it's impossible to know what the common ancestor of a eukaryote is, because we know it's a mixed bag of many parts. We can figure out what your common ancestor is, but we can't figure out what the common ancestor is of a eukaryote because eukaryotes came from things that could swap DNA. No, eukaryotes evolved from prokaryotes that could swap DNA. So it's, wait, sorry, you're asking me if the eukaryotes can swap DNA? Yeah. Okay, we can't. I would imagine there are probably some eukaryotes that can, but most can't. Most can't. Okay? But bacteria definitely can. So, no, what I'm trying to tell you is that when you're trying, because we've been talking about how do you reconstruct the common ancestor of humans and chimps, right? What would you do as a mental exercise? How would you do that activity in your head? Yeah, but then you would also look at, okay, what do they have in common, right? What do humans and chimps have in common, right? Or if you want to know what the common ancestor of mammals are, you have to look at what do they all have in common, right? So, for example, what would the common ancestor of a mammal, what characteristic would it have had? For a mammal? It would have been hairy, yeah. Would it have full body hair though? Okay, but it would have had hair. What else? Quite a few. Specialized, right? Does it make milk? Now, how do you know it has these characteristics? Because all mammals do, right? So when you're reconstructing a common ancestor, you can do the exercise in your head, right? Problem is, when you're trying to figure out what was the last common ancestor that gave rise to eukaryotes, it's very hard to do that activity in your head because it wasn't one thing. It wasn't one organism that gave rise to chimpanzees and humans. It's many things that gave rise to eukaryotes. Remember, we're a chimera? Remember what chimera means? Right, it's a combination of things. Okay? So when we talk about, and this is my last point, when we talk about like, here is a human, right? And here is a chimp. And they have a common ancestor. We think of that common ancestor as something, a particular thing, right? We don't know what it is, but it's one thing that eventually splits, underwent, what's that process when something splits and becomes new species? It's called speciation, right? But we think of one thing. When you think of the common ancestor that gave rise to eukaryotes, so that would be this common ancestor, it's not one thing. It's many things that contributed to it. Okay? So that's it for prokaryotes. I hope I confused the hell out of you guys. And that is